Hello, I'm René Hägerling and I will now talk about 3D visualization of the vasculature in human and rodent tissue samples using light sheet imaging. If we talk about the vasculature, especially in the context of lymphovascular diseases such as lymphatic malformations, GLA or GSD, we have to keep in mind this term represents a very heterogeneous group of vascular alterations affecting various parts of the body, for example the head, the upper or lower limbs, organs, or even the entire body. If we now want to identify the underlying cause of the disease, we can of course perform a genetic testing to elucidate the genetic alterations causing the phenotype. But we can also perform a histological analysis, which I will now focus on. For this purpose, we will use a tissue biopsy or sample which were obtained from surgery or a simple punch biopsy. And according to the current gold standard in 2D histology, the sample is embedded, sectioned into very thin sections, transferred to glass slides, stained and finally visualized under a fluorescent microscope by researchers or pathologists. And based on the 2D analysis, the underlying vascular phenotype is described. And we have performed such an analysis also for our showcase here. We have performed an H&E staining where you can see luminal structures, most likely vessel structures, and have also stained for blood and lymphatic vessels using specific markers. However, based on this data, it is not possible to describe the vascular phenotype in very much detail. We do not know if the underlying phenotype is a disrupted lymphatic network, if there are issues in terms of vessel specification, or if it is even a hyperplasia or a similar phenotype. So we do not know. And the reason for this is shown here. The interpretation of two-dimensional sections is very difficult due to the limited 3D information. I want to show you this using this cartoon on the right hand side. If you compare 2D sections in D&E, the sections look identical, but 3D reality is much different. In D, both lumina are parts of one vessel, where in E, both lumina are part of two separate vessels. Therefore, we cannot make clear statements on the vascular anatomy based on the 2D sections. In addition, 2D histology is always associated with tissue loss and tissue distortion, which even makes the interpretation more difficult. Therefore, we came up with a solution: optical sectioning using a light sheet microscope in combination with three-dimensional reconstruction. I always compare this technique with a CT scan of a biopsy, but we use, instead of X-rays, fluorescent light. And by capturing several thousand optical sections, we can analyze and visualize the entire sample like in a CT or even angio-CT. This is an example of the blood vasculature of a healthy control sample. You can nicely see the vessel architecture, the blood vessels in white, and in a few seconds you can also see the lymphatic vessels in red. And I'm sure you will agree with me that using this approach you can easily understand the 3D architecture of the vasculature in this healthy control tissue. We then used the same approach for patient samples and visualized a lymphatic malformation showing the big difference compared to the control on the left side. You can see the kind of chaotic vessel organization which can assist you by choosing the most suitable pharmacological treatment for this patient. For example, in this case, it is quite likely that a patient with such a vascular phenotype would benefit from sirolimus treatment, because sirolimus would cause or will cause a vessel normalization, whereas a patient with a vascular phenotype like here on the right side will most likely not benefit from this treatment, because immunosuppressive drug will not support reconnection of vessel fragments. So using this approach, we are now able to identify the underlying phenotype, in this case a hyperplasia of lymphatic vessels. As we want to go one step further and not only perform a qualitative but quantitative analysis, we've developed an unbiased analysis algorithms for quantification in three-dimensional space, in this case of the blood vasculature. Here you can see, for example, a 3D reconstruction of the blood vasculature. Then we perform a vessel mass extraction followed by segmentation like in an angio-CT, and finally a quantification of specific parameters we're interested in, like in this case for the vessel segment straightness. But how do we obtain such 3D videos? For this purpose, I want to briefly go through our sample preparation pipeline. We always start our sample preparation with an immunofluorescence staining. Therefore, we stain our targets of interest, for example, blood and lymphatic vessel. As standard IgG antibodies are very big, penetration of tissue is quite complicated and time-consuming, of course. And especially for bony tissue, IgG antibodies even fail to penetrate the entire sample. Therefore, we have developed new protocols involving a new class of antibodies, so-called nanobodies. Nanobodies are derived from alpacas or llamas, or even sharks and crocodiles if you prefer, and are very small in size, only 10% the size of standard IgG antibodies. And as tissue penetration correlates with molecular size, nanobodies can penetrate the tissue much faster and can even penetrate bones. This means you can prepare your sample not within weeks, as you would need for staining with standard IgG antibodies. Now you can prepare your samples within minutes or even hours. And this is one example of our first nanobodies. In this case, a nanobody for blood vessels, and you can nicely see that all blood vessels in this skin biopsy are visualized and therefore allow 3D reconstruction of the entire blood vasculature using this nanobody. And after staining, you have to make your sample translucent, because otherwise the sample cannot be optically sectioned, because light cannot penetrate a non-transparent tissue. Therefore, we perform optical clearing of our sample. That means we make it translucent, and this you can see here on the left hand side. So here are skin biopsies of a human patient on the left side before clearing, here after clearing, and you can easily see that you cannot see the sample anymore. This means it's perfectly translucent. And this works also for rat or human bone tissue, which is shown here on the left side before clearing, on the right side after clearing, and now you can see in this rat bone easily the blood cells. And finally, there is the optical sectioning. For this purpose, I've included a commercial video from Zeiss which illustrates the process really nicely. So that's the imaging cuvette where you place your sample in. And here you have your specimen of interest. In this case, in this demo video, it's a zebrafish embryo. You now insert this sample into the light sheet microscope. You place it into the right position, into the position you need for imaging. And once you have finished the placement of the sample, you can directly start imaging. And this is illustrated here. So we have here our light sheet, and using the light sheet we capture hundreds of optical sections, like you can see on the right side. And after capturing all optical sections, you can then perform the 3D reconstruction of your structure of interest, in this case the vasculature of the zebrafish embryo. We can not only visualize human tissue samples, but are also able to visualize mouse and rat tissue samples. For example, here on the left hand side you see a mouse embryo, on the right side intestinal lymphatic vessels of a mouse, and in the middle embryonic mouse heart. And with this slide I'd like to sum up my presentation. I hope I could convince you that light sheet imaging allows visualization of the entire vasculature in mice and men, that it allows us to get new insights into pathogenesis of lymphovascular diseases, therefore provides an improved phenotyping and identification of new subtypes, or even a new classification based on 3D histology, that the 3D histology offers some kind of decision support for pharmacological intervention, for example sirolimus treatment, which I explained to you a few moments ago, and of course the 3D histology will be and can be an essential part in precision medicine for lymphovascular diseases. And for that, I'd like to show you, as a very last slide, our outlook on what we are currently working on at the Charité for patients with lymphatic malformations. So we have set up a new study and have already started recruiting these patients, and hope within the next 2 years we have a successful evaluation of this concept, and hope that we can establish a new innovative patient care concept for patients with lymphatic malformations in Germany. And for this study we combine the 3D histology, which I introduced to you a few moments ago, with molecular genetics, meaning we perform sequencing of blood samples as well as tissue samples, and then in the next step we will also extract endothelial cells from the tissue biopsy, culture these cells, have a primary cell culture of our patient tissue, and then perform an FDA-approved drug sublibrary screening based on approximately 100 FDA-approved drugs. And this would then offer decision support for our colleagues and pediatricians and pediatric surgeons to support treatment of these patients. And this brings me to the end, and I'd like to thank you for your attention, and I'm happy to answer your questions. Thank you.